dear rector, dear colleagues, dear Anneline, dear friends. It's a great honor for me to receive this degree here today. I'm very happy to be here in this country also, which is one of the ones with the highest densities of Neanderthals in the world. There are so many sites, Spy, Goyer, etc., here, some of which we have had the pleasure to work on over the years. And what I wanted to do is to then describe a bit to you our efforts to sequence the Neanderthal genome and the Denisovan genome, and then spend most of the time to discuss how genetic variants that come from these extinct forms of humans have an influence in people today, and end by talking a little bit about what might set, or how we go after things that might set modern humans apart from Neanderthals. But let me just then, before I do that, remind you about what I think all of you already know, that if you study the human genome, if you sequence human genomes from around the world, an interesting observation is that most genetic variation exists in Africa. In spite of the fact that, of course, there lives more people outside Africa than inside Africa today, the genetic variation outside Africa is less. And the reason for that is then that there is a sort of component of the genetic variation in Africa that's unique to Africa. And the interpretation of this is that the ancestors of modern humans, the ancestors of everyone alive today, emerged in Africa, lived there for a while, accumulated genetic variation, and part of that genetic variation, so to say, went out and colonized the rest of the world. And by genetic tricks, we can also figure out approximately when that happened. And it's quite recently in evolutionary terms. It's sort of less than 100,000 years ago or so. And the fascinating thing for me is that at that time, say 100,000 years ago, 50,000 years ago, modern humans were not alone on the planet. There were other forms of humans inside Africa and also outside Africa. And most famously then in Europe and Western Asia, there were the Neanderthals. And in Eastern Asia, there were other forms of now extinct humans that we begin to learn a little bit about now. So why are we so interested in Neanderthals then in our group? I would say that there are several reasons, but primarily perhaps two reasons for that. The first reason is that the Neanderthals to the left here are the closest evolutionary relative of everyone alive on the planet today. So they appear in the fossil record something like 400,000 years ago and live until approximately 40,000 years ago when they disappear, generally in connection with that modern humans appear on the scene. And since they are our closest evolutionary relatives, it's sort of them we should compare ourselves to if we want to ask from a genetic or biological perspective what's unique to modern humans. So in what ways are we different from them? In what ways are we similar to them? And another sort of fascinating thing is that they were here quite recently, until 40,000 years ago or so. So until 1,200, 1,400 generations ago, there were Neanderthals here. So the question is what happened when we met? How did we interact with each other and so on? But to then ask that from a genetic perspective, you will need to retrieve DNA from bones that are at least 40,000 years old. And then that then, as you heard in the introduction, goes back to work in the 80s. We started looking at ancient Egyptian mummies, which are just 3,000-4,000 years old, look very well preserved, like this example of a actually 2,300-year-old mummy here. If you look in the microscope in the tissues of such mummies, they do look quite well preserved. So basically in the skin here, you can see structures that are cell nuclei where DNA should be preserved. So I was very enthusiastic at that time. I started extracting DNA from mummies like that, cloned it in bacteria, and showed that there was some human DNA among the DNA one had extracted, and published it to great fanfare. The sort of sad thing is only that over the coming years, then it became more and more clear that those DNA sequences I had published there probably didn't come at all from the mummies, but probably from myself or some museum keeper or some archaeologist or so. Because what one didn't realize was that there are many technical problems with this. And they sort of come from the fact that there is very little DNA generally that survive in such tissues. So if you compare from fresh tissues and these ancient things, often 100,000-fold, a million-fold less DNA there. It's degraded to short little fragments. There are chemical modifications there. And the vast majority of the DNA doesn't come, say, from the mummy, but from microorganisms, bacteria, fungi that have lived in the tissues. And that then means that tiny amounts of human DNA that's in an environment, on a dust particle, for example, which you wouldn't at all influence your experiments here, may totally overwhelm the experiments when you study ancient tissues, which was what had happened there to me. So over the next few years, we sort of progressively worked more and more on sort of avoiding this contamination problem by sort of clean room conditions and lots of different measures. We worked on animal remains because it's then much easier to distinguish endogenous DNA, say in a mammoth should be elephant-like, than in a human remains where it's much harder. Improved all the steps in how you extract the DNA from the tissue, handle it, these sort of short modified fragments and so on. We got much, much better at that. Sequencing techniques got much, much better. And we learned things too. For example, when you then look at these fragments, we learned that if we look at the ends of the molecules, at the ancient molecules, we find typical chemical modifications. So sort of we find that certain bases, a C, appears as T's towards the end of the molecules. And that's due to a sort of hydrolytic damage that happens in the DNA where the cytosine loses its amino group, becomes uracil, and uracil is then read as T. So that was initially felt to be a big problem, that we sort of have errors in our DNA sequences, but it can also be an advantage, because this process takes hundreds and thousands of years to happen. So when you see these types of errors, you can also feel very sure that you have an ancient DNA molecule. So with all these sort of advances and these tricks, we sort of felt in the beginning of this millennium that one could come back and try to look at Neanderthals then. So part of that involved looking through many archaeological sites to find ones where best preservation of DNA. We focused in the end on one site in Croatia, Vindija Cave, and 3 different Neanderthal bones from different individuals at that site. Sequenced over 1 billion DNA molecules, sort of taking into account these errors towards the ends there, and mapped them to the human genome that had then become available. So by 2010, we had a little over half the Neanderthal genome seen by at least one such fragment. And we could begin to ask questions. And one of the first questions we sort of had was, what happened when modern humans met Neanderthals? Did one mix with each other or not? Which was a long-standing debate in human evolution. So an expectation, if Neanderthals would have mixed with modern humans, would of course be that Europeans today would carry more Neanderthal genetic variants than would share more variants with Neanderthals than people in Africa where there had never been Neanderthals. So to then ask that question, we put together a consortium of different people, population geneticists particularly, illustrated here, and we then compared the Neanderthal genome from Croatia there with 5 genomes from around the world that were sequenced with very similar technology to have the same error rate everywhere and so on. And then we asked a very simple question. to just illustrate this, we start with 2 African individuals here. So we look at these 2 present-day people, find all positions in the genome where they differ from each other, and then say, the Neanderthal genome now, how often does it match that African and that African individual? Is there a difference in matching? And there's no reason to assume that a Neanderthal would be closer to one African than another African, right? Because there have never been Neanderthals there. And indeed, statistically speaking, there is no difference. We then did the same thing with a European individual. We debated for a while what's the most typical European individual, and that was obviously a French person, and an African individual. And when we now ask this, to my surprise at the time, actually, we found significantly more matching to the European individual than the African individual. Even more surprising, when we took a Chinese person and an African person, although most people would say there have never been Neanderthals in China, we find more matching. Papua New Guinea and Africa, where everyone would agree in Papua New Guinea there have never been Neanderthals, we see more matching. So the sort of idea that came out of that was then that when modern humans spread first through Africa, then out of Africa, they probably met Neanderthals, probably in the Middle East, where we know there were Neanderthals. If these early modern humans out of Africa then mixed there, they could sort of carry this Neanderthal component with them to other parts of the world where Neanderthals didn't exist. And the result is then that 1 or 2% of your DNA, if your roots are outside Africa, come from Neanderthals. So what has happened since then is then that we have now much better Neanderthal genomes that not cover just half of it, but everywhere where we can map these short fragments many, many times over. So we can more in detail compare Neanderthals to individual genomes today. So if we look on just one chromosome here, each line would be an individual today. Red are then fragments of 10,000 up to 100,000 nucleotides that are very similar or identical to the Neanderthal genome. You will see that everyone carries some of these. But you will also see that different people carry different fragments here. So although each individual carry 1 to 2%, we can jump from person to person and puzzle together longer pieces of the Neanderthal genome, as is indicated down here. And that adds up to at least 40%. Actually, now when we have hundreds of thousands of genomes, up to 50, 60% of the Neanderthal genome is still walking around on 2 legs today, so to say. Another thing that we then stumbled across early on was from this site in southern Siberia, the Niseva cave, where the archaeologists who excavated there had found this tiny little fragment of a bone, had been very skilled, I think, to recognize that this might come from a human. And when we sequenced the genome from this, it turned out to not be a Neanderthal, as we had thought it would be, nor a modern human, but something else that went quite far back to a common ancestor here shared with Neanderthals. So this sort of individual goes back to some ancestor shared with Neanderthals about 400,000 years ago or so. So we sort of realized we stumbled across this now extinct form of human, had to give it a name. So just as Neanderthals are called Neanderthals after the site where they were first seen, we call these individuals then Denisovans. We could ask if they had also contributed to present-day people. It turns out they have. We find no contribution in Europe or Western Asia, but wherever you look in Eastern Asia, it's a small contribution, less than 1%. But in some parts of island Asia, on Papua New Guinea, for example, is up to 5-6% of contribution from these Denisovans on top of the Neanderthal contribution, so to say. And not only that, the Denisovans seems to have been quite diverse and more numerous probably than Neanderthals. So for example, this is work from a group at Princeton that have analyzed our published genomes. So if we look, when they look in Papua New Guinea and plot here fragments in the genome that are more and more similar to the Neanderthal genome. They find this Neanderthal contribution here that's quite similar to our Neanderthal genome. Here is matching to the Denisovan genome, and they find this contribution in Papua New Guinea, but it's quite distant from the Denisovan genome we sequenced. If they now do the same thing in Japan, they find this contribution from Neanderthals close to our genome. They find this contribution from Denisovans quite distant from our genome, but in addition, a contribution from Denisovans that are quite close to the genome there from southern Siberia. So there are at least sort of two genetically distinct groups of Denisovans that have contributed to people in Japan and in China. So we can already see that there were sort of different subgroups of Denisovans, that all of them mixed with ancestors of present-day people. In spite of this, we sort of, until recently, had direct evidence of Denisovans only from this site in southern Siberia. But there is now another site in Tibet where one found this mandible some years ago, which turned out to be quite old. One have not been able to retrieve DNA from it, but there is one protein that has one amino acid sequence position there that looks like Denisovan. So a while ago, one was able to go back to this site and study archaeological sediments. So an interesting development in the field is that these methods are so sensitive, so one can now go and just take soil samples in a cave where people have lived and see what animals have been there and what humans have been there. So in this cave then, together with Chinese colleagues, one could sort of study DNA through the sediments here, and indeed find that in this cave were individuals living that were sort of related to this group of Denisovans in Siberia and other more distantly related ones there at other times. So we now have at least two sites where we know Denisovans have lived, and I'm very confident that actually many, many other sites in China, for example, will turn out to be remnants of Denisovans, fossils in museums and so on. So just to summarize this first part and what we think we know from studying the genomes of the origin of Neanderthals, Denisovans, modern humans, is that Neanderthals and Denisovans probably have some ancestor in Africa also that come out of Africa, evolve into what we call Neanderthals in Western Eurasia and what we call Denisovans in Eastern Eurasia. We don't know where the border between these groups have been and it shifted over time, but we do know that at site in southern Siberia there were Neanderthals at that site in the Denisova cave sometimes, and Denisovans at other times. Then there is an origin of modern humans in Africa 300,000, 400,000 years ago. They start spreading out of Africa and meet and mix with ancestors or with Neanderthals. And just last week there was a paper where they dated much better this major pulse of mixture to 49 to 52,000 years ago, so quite recently. And then modern humans continued to spread. They mixed several times with modern humans, with Neanderthals, and with Denisovans in the East. And then, of course, modern humans continue to spread to other parts of the world where hominin forms have never been, out in the Pacific, to Americas, and so on. And these other forms then disappear, but live on in this genetic contribution to people today. You may then ask, are people in Africa fundamentally different in not having this sort of contribution from other forms of humans? I don't think so, because modern humans obviously spread across Africa too. If one mixed with other forms outside Africa, one probably did it also in Africa. But since we have no genomes from now extinct forms in Africa yet, it's sort of hard to really prove that. But I think once we have that, we will find that that happened in Africa too. A fascinating thing is also that we're starting to get very direct evidence of this mixing by looking at very early modern humans in Europe particularly. So the first such case was in Romania, a site called Oase Cave, where cavers found this mandible some 10 years ago, which is a modern human mandible, but it's dated to 40,000 years ago, so it's a very early modern human. So we were, of course, very interested in if this person had already mixed with Neanderthals or not. And indeed, if we now look at the different chromosomes here, and in blue show fragments similar to the Neanderthals, you will see that they don't only have 1 or 2% this individual, but huge segments on the chromosome. Sometimes more than half a chromosome looks Neanderthal. So this, of course, means that this person in its family history had a Neanderthal individual. So we can then show that either 6, 5, or 4 generations back, this person 40,000 years ago in Romania had a Neanderthal ancestor. And I thought it would be like total chance that we would stumble across such an individual. But in the meantime, there's another site in Bulgaria, Batukiro Cave, where one finds technology typical of very early modern humans. Deep down in the stratigraphy, you find early modern humans 45,000 years ago. Later on, 10,000 years later, there's also modern humans there. And we were able to recover genomes from these very early modern humans down there. And it turns out that all of them actually have large chunks of Neanderthal DNA, having sort of Neanderthals in their family history. 10,000 years later, in the same cave, people look pretty much like people today. So in the meantime, just last week, there came a study from another group in our institute, where they had studied early modern humans from another site in Germany here, in Ranis. And it now turns out that there is sort of this northern group of early modern humans 45, 50,000 years ago in Europe here. They don't seem to have had this direct interaction with Neanderthals that can be detected. And there's more this southern group, the Oase and Batukiro, that I just showed to you, where all 4 individuals for which you have data have mixed with Neanderthals. So it seems that at least some early modern humans groups, when they came to Europe, mixed very readily with Neanderthals. So I think that sort of hints at that one reason why Neanderthals disappear may not necessarily be that one killed them or something like that, but that they mixed with probably larger modern human groups and sort of were just assimilated so that their morphology disappears, so to say. And indeed, there is evidence that modern humans were more numerous. So if we look at the amount of genetic variation in Neanderthals, it's quite small compared to present-day people here, suggesting that they were smaller groups. And there's also some archaeological evidence of that. If you look at sort of early modern human sites in southwestern France, for example, there are more sites there, and they also seem to have been more intensely used in terms of how much butchered animal bones you find, how much stone tools you find. So, but then what my group is most interested in, since the 5 years or so, is to actually use these genomes. Since we now have high-quality genome from our closest relative, we can sort of begin to look at where genetic changes happen. So when we find genetic changes that we share with Neanderthals, and they're not there in the apes, that's changes that happened here. When we find things only in Neanderthals and not in us, they happen on the Neanderthal lineage. And when we find things that everybody today shares and Neanderthals don't, they happened here. So I then want to give you some example of sort of such Neanderthal-specific changes and what we can learn about them, things that happen there. And the first example comes from an ion channel. So it's a protein that sits in the cell membrane and can let ions through into the cell. And this is Hugo Seeberg's work at the time at the Karolinska. He's very interested in ion channels. And he found that all Neanderthal genomes we had access to had Among them, there was this channel that had 3 amino acid changes, and no other protein actually had as many as 3 changes. And this channel here is an interesting one. It sits in the peripheral nerve endings out in your tissues and is involved in initiating the sense of pain when you hurt yourself. So what we then did was express this Neanderthal version of this channel and the modern human version in human cells, and then study when you have an electric stimulation of the cells, how much ions are let through the membrane. And we found that the Neanderthal version let through more ions, as if it was more sensitive to the stimulation. I'm not an electrophysiologist, but one could then show that that was not because the ion channel opens quicker, but once it opens, it remains open longer before it gets inactivated. And one could also show that this relies on 2 of these amino acid changes are necessary for this effect, and the third one doesn't seem to matter. So we thought that this was unique to Neanderthals, that no one carried this today. But another cool development is that we begin to have these big biobanks where we have genome information from hundreds of thousands of people, and also medical records and questionnaires they have answered, and so on. And one of the most accessible of those biobanks is the UK Biobank in Britain. And when we looked there then among 360,000 individuals, we found some that actually carry the Neanderthal variant due to gene flow from Neanderthals then. So we can then use these people as model organisms, if you like, to study the effects of this. So there were 17 questions that all these people had answers that had to do with pain, all kinds of pain, headaches, stomach pain, back pain, what have you. So you can look at what does sort of how much pain you experience in your life correlate with. And as you get older, it's very sad. The biggest correlation is simply with age. The older you are, the more you complain about pains and aches. It's sort of trivial in a way. You have more medical problems the older you are. But more relevant, carriers of this variant, they do report more pain in their lives. And if we should relate it to this age effect that we can perhaps sort of have a feeling for what it means, it is as if you were 8 or 9 years older in terms of how much pain you experience if you're a carrier of this Neanderthal variant. Now, that doesn't mean that we can necessarily say that Neanderthals experienced more pain in their lives, because we all know that the experience of pain is very much modulated in our spinal cord and particularly in our brain. Even the same stimulation can sometimes be painful, sometimes pleasurable in our lives. But it is still sort of interesting that all these people that report more pain here, they are heterozygous for this. They have the Neanderthal version on one chromosome and the modern version on the other, of course. Since this variant is so rare, so we haven't found anyone who has it on both chromosomes. Neanderthals had it on both chromosomes. So maybe we sort of have to at least be ready to sort of reconsider our view of Neanderthals as sort of insensitive, brutish people. Maybe they were actually overly sensitive and everything hurt when modern humans arrived. So another example then involves a hormone that I think many of you have heard about, progesterone. And in this case, we're interested in the receptor of the progesterone hormone. So the protein the hormone binds to when it is to exert its effects. And you may know progesterone has many effects, but a major effect is to be produced in the ovaries after ovulation and prepare the uterus for a possible pregnancy. And it was already known that there is a variant of the progesterone receptor that's associated with increased risk of premature births, and that came from Neanderthals. And if you look around the world, it has this typical distribution for Neanderthal variants where it's absent or almost totally absent in Africa, exists outside Africa, and it's then sort of puts the baby at risk, of course, particularly in a society where you don't have medical care. So it had been speculated that this was very bad for the Neanderthals and so on. But another interesting development now is that we begin to have thousands of modern human genomes distributed in time over the last 10,000 years or so. This particular group in Leipzig, Johannes Krause's group and David Rice's group at Harvard, has sequenced thousands of individuals since 10,000 years ago until present day. This is not quite up to date. Nowadays we have something like 20,000 such genomes. So what we can then do is to follow the frequency across time of such variants and see, have they decreased, have they increased, what has happened. So if we do this in a little animation here now for this variant, the progesterone receptor variant from Neanderthals, the Neanderthal genomes are in red, carriers of this are black, non-carriers of this Neanderthal variant are gray. We start 15,000 years ago and go forward in time, and around 7,000 years ago, you will see how this formally explodes in frequency, how the frequency increase a lot. So that seems to make no sense. Why should something that put your baby at risk of dying increase in frequency? So that made us go back to the UK Biobank again and ask what are the associations with these variants there actually. And we now look at it the other way. We look at the modern human variant, the non-Neanderthal variant here for the progesterone receptor. What we find is that the modern version, the non-Neanderthal version, is associated with increased of bleeding early in pregnancy, with increased frequency of miscarriages, and also negatively associated with the numbers of sisters and also brothers actually that you have. So if you have the modern version, you have a bigger risk of miscarriages, and you actually have less brothers and sisters if you carry it, because it's a big chance that your mother also carried it, of course. So the situation seems to be that this Neanderthal variant, yes, it is associated with increased risk of premature birth, but it also protects against miscarriages earlier in pregnancy and actually results in more live births. So it seems to be a sort of trade-off where this Neanderthal variant saves pregnancies that would otherwise be lost. And the price you pay, so to say, is that these babies are more at risk of being born early. And we begin to understand why too. Because if these Neanderthal variants here, if you carry the Neanderthal variant, you actually express more of the hormone, of the receptor, sorry. You have more of the receptor, so you could imagine you have sort of more of a progesterone effect if you have more of the receptor. And indeed, there are now studies where one gives progesterone, gives the hormone to women who have experienced several miscarriages, and you can significantly increase the number of live births. So this seems to be a case then where it's actually quite good to carry the Neanderthal version of a gene. And this is sort of typical. There are many genetic variants around in the population that are known since a long time to be medically important that turn out to come from Neanderthals. So an example here is these two enzymes that sit close to each other on one chromosome here that are expressed in the liver and are involved in metabolizing many drugs you take. And variants of these clearly come from Neanderthals here, are very close to the Neanderthals. So if you, for example, take ibuprofen for pains, and if you're a carrier of the Neanderthal version, you have a much better effect because this enzyme digests it slower, so you have much longer, higher half-life in your blood than if you're not a Neanderthal carrier. But more seriously, if you are a carrier of these enzymes and take warfarin to prevent blood clots, you need to take a lower dose in order to avoid side effects that are other kinds of bleeding that could be very dangerous. So a biggest example for me of sort of influence from the Neanderthal gene flow came in the pandemic. As you know, almost everyone got infected with this virus. Most people got hardly any symptoms or very mild symptoms, but some people became severely ill and some even died. We know some risk factors: old age, male sex, diabetes, what have you. But all these risk factors are not enough to explain why some people became so very ill and some hardly got ill at all. So it was quite clear early on that there must also be genetic factors in us that sort of determine how badly we're affected by this virus. So we were peripherally involved in a big international consortium in 2020, looking for such genetic risk factors. And in the summer of that year, when the first results were in, the results were actually quite striking. There were on chromosome 3 one piece of our genome that seemed to be very important for how sick you become when you have this, when you're infected then with SARS-CoV-2. And even more shocking for us was that when one looked at these risk variants there, they were clearly coming from Neanderthals, particularly from this Croatian, very close to this Croatian Neanderthals that we had sequenced. Whereas a protective variant that put you less at risk of becoming severely ill was sort of modern, if you like. So clearly this risk variant has sort of come over from Neanderthals and exists in some of us today. And it's quite a big genetic risk factor. So if you were hospitalized early on in the pandemic in Scandinavia and were not for severe COVID and were not a carrier, you had a 7% risk to die from the virus. If you were a carrier, your risk was something like 13% or 14%, about twice as high. So it's of course very interesting what hides in this region here on the chromosome. Unfortunately, I should say, it's a very complicated region. There are many genes there. Many of them are influenced by this risk factor in their expression in different tissues. There are at least 3 sort of hypotheses around for this. So we and others are still working on this hard. Of course, if we understood why this confers bigger risk, we might be able to treat severe COVID also much better. But you can then use the frequency of risk factor around the world, typical for Neanderthal things absent in Africa, existing outside Africa, to estimate how many extra deaths did we have in the pandemic so far due to this Neanderthal contribution. And it's at least 1 million extra deaths that we've had due to this. You can also see that there are some interesting patterns here, that this variant is absent or almost absent in China and Japan, for example, whereas it's very frequent in South Asia and India, Sri Lanka, Pakistan. So there's clearly something that has selected against it in East Asia. We don't know what that is, perhaps corona pandemics in the past. But there are also things that have made it good to carry this thing in South Asia. We also don't know what that is. But we are beginning to learn that this region influences outcomes also in other infections. So Hugo Seeberg again noticed that if you look at this risk factor here, if you look 1 million base pairs away on the chromosome, there's a famous gene there that you may have heard about, CCR5, which is a co-receptor for HIV, so the virus that gives you AIDS. And it turns out that if you carry this risk factor for COVID, you express less of CCR5 in your tissues. And as you know, if you express no CCR5, if both copies on both chromosomes are knocked out, you're actually resistant against HIV. The virus cannot infect you. So it then turned out, and we looked in big cohort studies, that indeed, if you carry this Neanderthal risk variant and are exposed to HIV, you're around 25% less at risk of becoming infected. So this risk locus on chromosome 3 is a risk locus for severe COVID, but if you're exposed to HIV, it actually protects you to some extent from becoming infected. And it illustrates, I think, that genetic variants almost always have both good and bad effects depending on the environment, in this case, on which virus you're exposed to. So they're a sort of double-edged sword, if you like, or there must have been other things that this influenced in East Asia and South Asia, so sort of multi-edged swords, right? And before now, leaving the Neanderthals and COVID, I should say that when one now has done much bigger association studies of severe COVID, one has found other loci where Neanderthal variants are actually protective. The sad thing, the effect size is much, much smaller. So whereas this thing doubles or triples your risk of dying, the protective variant from Neanderthals reduces it by 20% or so. So before coming to the end, then I want to give you a little bit of feeling for what we are particularly interested in maybe in our group now. And that are then this type of changes. So not the changes we discussed so far that happened on the Neanderthal lineage and came over to us, but things that changed on the human lineage since we separated from Neanderthals and are not there in the Neanderthals. And we're so interested in these changes because we imagine that some of the genetic background for functions unique to modern humans may hide among these changes. And I do think that can be debated, some of this, but I do think that modern humans are rather special. So technology and culture in modern humans start changing very rapidly, at least 70, 80,000 years ago, compared to Neanderthals, Denisovans, any other forms of extinct hominids. Figurative art, art that really depicts something, also comes with modern humans. There are some things that are presumably Neanderthal art, but I generally like to say that that looks like very modern art to me because I can't see what it depicts at all. Whereas things like this, that you see what it is, comes with modern humans. And modern humans, as we discussed already, become much more numerous than Neanderthals or Denisovans. They also start spreading systematically across open water, for example, where you don't see land on the other side that other hominids have never done. So there is something with behavior, culture or so that sets modern humans apart, I think, and then made us become today billions of people. So an idea is that sort of some of the readiness to accept sort of assimilate this culture or so may hide by the changes that happen there on the line to modern humans and exist in all of us today or almost all of us today. So we sort of can list all these things and look at them, and they are not that numerous. It's in the order of 30,000 changes across the whole genome. So whereas between two genomes in us here in the room, there's something like 3 million differences. But if we make the requirement that we should, almost all of us should carry something and Neanderthals should not have it, then it's just 100 times less or so. And they affect then, have this bias then, that we are most interested in things that may influence behavior or sociality or so may influence the development or function of the brain. And we have been a little lazy focusing on those changes that change amino acids in proteins, since in some sense they are easier to study. So you can list all the genes encoding proteins that have such changes. We've studied sort of 12 or 13 of them so far. I just want to give you a flavor of this sort of studies by mentioning one of these studies, because Annelina, that you just heard from, has worked on that in Dresden. That's an enzyme called TKTL1, and she worked on that in Wieland Huttner's group and a bit with us in Leipzig. And it's sort of involved in energy metabolism. And one approach to then study this is that you can take human stem cells. Stem cells are then cells that can differentiate into other tissues. And you can use sort of CRISPR-Cas9, this method of changing particular positions in the genome. And in this case, we then change that position back to the ancestral Neanderthal-like state. And you can then use such stem cells that you have modified to look like Neanderthals for this one position to make brain organoids, which are little sort of balls of a few millimeters that you have in tissue culture that mimics early stages in brain development. And when then Anneline looked at this, in such brain organoids, in the epithelium here, where stem cells divide down here, migrate up and form neurons, she found something very interesting, that if she looks on the stem cells there in that part of the epithelium, look in the ancestralized organoids, there seem to be less stem cells there. And that also seems to have consequences, because if she looks at the neurons up on top there are fewer neurons formed. So this looks very interesting. Neanderthals, of course, had just as big brains as we had, but maybe there are then more neurons formed in a modern human brain than in the Neanderthal brain. But I think there are also a number of sort of caveats we should have when we look at this. So sort of big question in the room from this study and from other studies is then, if neurogenesis differs in modern humans from Neanderthals. But sort of the caveats I want to mention is that what we study here now is really early steps in brain development. And it could well be that these changes early on are compensated later on during embryonic times or even after birth, and may have no consequences in the adult then. Another is that sort of interesting traits like this are often influenced by very many genetic changes. So there may be many other changes, even in the opposite direction perhaps, that may influence such things. And as we have stressed again and again here, there are also modern humans, also for this variant, that carry the ancestral variant, and as far as we know, they are functioning just as well as anybody else. So there is sort of a struggle to understand what makes modern humans special. And to end, I would perhaps just take a step back to then say, how should we think about what it means from a genetic perspective to be a modern human? And we can perhaps think about how we think about Neanderthal morphology. We, of course, know that we have found a Neanderthal skeleton when we find it, because it has a combination of features. It's very robust. It has this bump in the back of the head, an oblong cranium, big brow ridges, and so on. But we can find people walking around on the street today that have just as oblong a cranium or just as big brow ridges as a Neanderthal. But no one walking around today have a combination of all these things. So it's really a combinatorial view of defining both the modern human skeleton and the Neanderthal skeleton. And we probably have to extend that also to other things, such as metabolism, brain development, and so on. That is a combination of features. So the suggestion would then be that you are a modern human if you carry most of genetic changes and whatever traits they encode, but none of these things are in themselves necessary to be a modern human, because there are many of these changes, probably many influencing even the same traits. That doesn't mean that there's not some of them that are more important than others, and that is in a way what we are struggling to try to find out in the next few years. So with that, I then hope I convinced you that it can be interesting to have the genome of our closest relative, because we can find these things that are unique to modern humans, unique to Neanderthals. We can study how things have come over from Neanderthals and Denisovans and influenced to us today. And the way we do that is then particularly these big biobanks, where you can associate genetic variants with traits, but also modifying stem cells, making brain organoids, for example, and sometimes even modifying animals to be more Neanderthal-like or modern human-like for certain genes. Most of this work goes on in Leipzig, in Germany, and to some extent then in a small group in Okinawa. And with that, I then thank you for your attention and wish you a Merry Christmas.